All right, guys, good morning. We're going to get started just a minute early, talking about a little bit of housekeeping stuff. So as you all should know by now, we are going to be having your first test on Wednesday. Your test is going to cover whatever we cover in class. I'm going to be going through some stuff fast today to make up some time. I'm going to be skipping things. Just follow along with me. Whatever we skip is not going to be on the test. You don't have to worry about it, okay? If you forget what we skip, go back and watch the recorded lectures. They're there for this exact reason, okay? So yeah, any slides that I put X's through or any materials that I say we're not going to talk about, those will not be on your test. Your test is going to be in person, like we talked about before. It is all on paper, so you don't have to bring anything other than something to write with, preferably a pencil, but a pen will work in a pinch if you don't have one, but preferably a pencil. I have special Scantron sheets that I use. They're custom printed ones, so I will hand those all out to you. Your test does have all of the choices, so it's multiple choice, true, false, short answer, matching, all the things. So there'll be a little bit of everything, but there will be short answer questions, so just be prepared for that. All right. Judging by, I have to say I'm quite surprised. I was looking at your answers. Judging by your answers, it appears that about 40% of you actually want to have class on Friday. So since that happened, I will probably still have class on Friday. Now keep in mind that if you don't want to come to class because you want to go home early, don't come to class. You're going to miss one attendance day. It's really not a big deal. It's not going to affect your grade. But we will have class. I will be taking attendance. Again, it's not that big of a deal. If you had plans to go home, I will be providing a recorded lecture, so it's not the end of the world if you don't want to come or you can't come. I was just going to cancel it if there were going to be like 3 people in this room. But there are a fair few of you that say that you actually would like to have class, so I will do that for those people. I think that's it. Are there any questions about the test in general? Or what you need to bring or anything else? All right, don't forget that on Wednesday, your 1 through 5 Connect assignments are also due. Those are due Wednesday by 11 a.m. By the time you walk into this test, those Connect assignments are due. And again, remember, they take about 40 minutes apiece, 40 minutes to an hour apiece. If you've been keeping up with it, it's not too bad. But if you've saved them all to the last minute, it's going to be a real bummer. But you still have time to catch up. And let that be a lesson for the next time. Okay? All right, so we are going to jump in right where we left off. And we are talking about the structure and the individual structures within a eukaryotic cell and how those cells differ from prokaryotic cells. And the biggest difference, or one of the biggest differences between prokaryotic and eukaryotic cells is that eukaryotes have things called organelles. And organelles are basically, they're named small organs. All right, so they're just little structures that exist inside of a eukaryotic cell that serve specific purposes. Like the organs inside your body serve a specific purpose, right? Your kidneys filter your blood and your liver produces proteins, all of these different things. We have organelles inside of our individual cells that serve a specific purpose. And if you've taken high school biology, all of this should be relatively familiar. We're not going to go into anything too deep here. It's just the basic idea of what these structures do and why they exist. So the first one is the endoplasmic reticulum. This is our first organelle. And the endoplasmic reticulum, I will refer to from now on as the ER, just because it's easier than saying endoplasmic reticulum. Okay? And they are a series, the endoplasmic reticulum as a whole is a series of flattened membrane tunnels. And the most important thing is that they are kind of attached to the side of the nucleus. Okay, so they're actually attached to the nuclear envelope. And they're kind of an extension, actually. So it's an extension of the nuclear envelope. So it's attached right to the side of the nuclear envelope. And the overall purpose of the endoplasmic reticulum is synthesis. And synthesis is just a fancy word for making stuff. Okay, the ER is mostly responsible for making the things that a cell needs. Now, there are two types of endoplasmic reticulum, which I'll show you in the next slide, and they each have their own purpose. But overall, their purpose is synthesis. And every single eukaryotic cell is going to have an endoplasmic reticulum. All right, so our two types of endoplasmic reticulum are the rough ER and the smooth ER, also called the RER and the SER. And the rough ER is called the rough ER because you can see the physical structure of the rough endoplasmic reticulum looks bumpy. When you look at it in this special electron microscope, you can see the straight lines. I'll try and do it in orange instead. The straight lines are membranes. Okay. The straight lines are membranes, and then attached to those membranes are a bunch of little dots. Okay, you can see all these little black dots I colored them in purple. And those little dots are ribosomes. Okay. So the rough endoplasmic reticulum, the membrane is embedded with ribosomes. It has ribosomes attached to the membrane. Now, does anybody know what ribosomes do? Do you remember? We talked about it a couple, like maybe last time. Yeah, they make proteins. So the rough endoplasmic reticulum, because it has those ribosomes, is responsible for making proteins. All right, so the rough ER is called the rough ER because it looks bumpy on a microscope. It looks rough. And it looks rough because it has all those ribosomes. The ribosomes are there to synthesize proteins. Okay, the ribosomes are the actual mechanical structure that forms proteins. We're going to talk more about that, how that happens later, but for now, that's what you need to know. And the smooth endoplasmic reticulum is called that because it appears smooth. As you can see, it's just a series of smooth membranes. There's no black ribosomes studded in the membranes. And so there's no ribosomes here. And so smooth endoplasmic reticulum is going to be responsible for making the things that don't require ribosomes. So of our macromolecules, right, we have proteins. What other ones do we have? Lipids and carbohydrates. Okay. So the smooth endoplasmic reticulum is going to synthesize carbs and lipids for the most part. There are some exceptions, but we're just talking about in generals right now. Okay. So the rough endoplasmic reticulum, where there are ribosomes, is responsible for synthesizing proteins. The smooth endoplasmic reticulum, no ribosomes, smooth surface, is responsible for synthesizing lipids and carbs. All right, so the endoplasmic reticulum, like I said before, is mostly responsible for making stuff, putting stuff together. Our next stop in kind of this set of organelles is something called the Golgi apparatus. And the Golgi apparatus is another series of flattened discs that are near the endoplasmic reticulum, but not usually attached to it. So it is not attached to anything, but it is not attached to the ER. So the endoplasmic reticulum, remember, are attached to the nucleus, and then the Golgi sits kind of separate from those two things. It's not attached to anything. Now, the Golgi's purpose, for the most part, is processing and distribution. Okay. If we think about the inside of a cell as like the inside of a factory, okay, the nucleus would be where all the information is kept, right? So the nucleus would be where the instructions are to make whatever you're making. Say you're making, I don't know, clothing, okay? You're making an item of clothing. You make t-shirts. That's what you do. Your factory makes t-shirts. Okay, the nucleus would be where all that information is kept. the files that have all your screen prints on them, the information about sizes, all the information is going to be kept in the nucleus. The nucleus is going to send that information to the assembly line, which would be like the endoplasmic reticulum. And all that information will say, I need 1,000 t-shirts in red and 1,000 t-shirts in green. I need this many to have long sleeves, this many to have short sleeves, whatever it is. And the endoplasmic reticulum is the assembly line. They're where those things are going to be physically made. Okay, so that information is sent to the endoplasmic reticulum. It is going to be physically made in the endoplasmic reticulum. It's where your t-shirts will be printed. All of the things will be manufactured or synthesized. And then the last step in any factory is to then process, package, and ship all of the things you just made, right? So you're inside of a factory, you just made 1,000 t-shirts. You have one company in Seattle that needs 250 and another company in Minnesota that needs 200 more. Whatever it is, your shipping department is then going to package those up the way they need to go. If any last minute details need to be fixed, they're going to slap shipping labels on it and then ship it wherever it needs to go. That is the Golgi. The Golgi apparatus is the shipping department of the inside of a cell, in the factory that is a cell. Okay, it's where all of the final changes are made, and we're not going to talk in specific about what that means. You just kind of have to know it as an idea, right? It's where any final changes are made, final processing happens, and then the Golgi is responsible for telling whatever was made where to go. Because whatever is made, whether it was a lipid or a carb or a protein, is going to have a specific destination. And it's the Golgi's job to package it up and send it where it needs to go. Okay? So the nucleus is the information center. The endoplasmic reticulum is the manufacturing center, or the assembly line, the synthesizing line. And the Golgi is the shipping department. All right, we're not going to talk about these. Skipping it. All right, so this is just another view of what I was just talking about. That in nature's assembly line, things start within the nucleus. This is where all the information is inside the nucleus. That information gets passed to the endoplasmic reticulum, where things are made. And then the things that are made move to the Golgi apparatus, where they are processed and shipped. Okay. And then those things will go wherever they need to go. They'll either go to the outside of the cell or to other parts of the inside of the cell, but we don't need to talk about that right now. Our next organelle, which is kind of separate from the other three, not the other three, the other two, I guess. The endoplasmic reticulum and the Golgi kind of work together with the nucleus. There's an assembly line there, like we just talked about. The mitochondria kind of sit separate from those. The mitochondria is responsible. Everybody knows this one, right? The mitochondria is the powerhouse of the cell. The mitochondria is responsible for producing energy that the cell needs to do whatever it needs to do. All of that information sending, manufacturing, synthesizing, shipping, processing, all of that requires energy. Every single step requires some amount of energy, and the mitochondria are responsible for producing it. Okay. Mitochondria do something called aerobic respiration. This is how they produce energy. And the way aerobic respiration works is the mitochondria takes sugars and turns that into oxygen and ATP. And remember, ATP, we talked about it briefly, is the energy currency. Okay, those phosphate groups act like energy currency. We'll talk more about ATP, I think, in the next module. But that's all you have to know for now. And we're going to talk more. This is aerobic respiration. Aerobic respiration requires oxygen, okay? Aerobic, aero. When something is aerobic, it means that it requires oxygen. We're going to talk about some organisms later that are what we call anaerobic. What's that? You guys look confused. What's that?
Is it convert sugars to oxygen in ATP or sugars and oxygen?
Nope, you're absolutely right. I messed it up. I put my thing in the wrong spot. It requires sugars and oxygen and converts that into ATP, which is a big difference, right? So it takes sugars and oxygen. Sorry about that. I messed it up. But this is the correct word, and the wording is correct. That's how she caught my mistake, is that the wording is correct. I just wrote it down wrong. So you take oxygen and sugars, and those two things together make ATP. Sorry about that. Okay, so this process does require the input of oxygen. We will talk about bacteria later that don't require oxygen input, and they do a different process. But cells that have mitochondria do aerobic respiration. Okay, and we're actually just going to skip the cytoskeleton. All right, so now that we have an idea about the basic structure of a eukaryotic cell, we've talked about all of the things that they have, or all of the things that all of them have. We've kind of skipped over some of the smaller things, but we have an idea about the basic structure. Now we're going to talk about the individual types of eukaryotic organisms that we are talking about in this class. Now remember, there are a lot more eukaryotes than we will talk about. We are specifically talking about 3, which I talked about at the beginning of this chapter. We're talking about fungi, protozoa, and helminths. And we're talking about those because they are microscopic and pathogenic. But I don't want you to think that these are the only eukaryotic organisms or even the only eukaryotic microscopic organisms. We're just focusing on these 3 because they tend to affect human health, these 3 groups. All right, so our first group are the funguses. And I also think it's important to note that we're going to talk about a few pathogenic fungi, but for the most part, just like everything else, the vast majority of funguses or fungi are neutral or helpful to humans. We're going to talk about the few that are not. Okay, but fungal cells come in 2 basic forms. And just as a reminder, we had it a couple slides ago, but what is the cell wall of a fungus made out of? Anybody remember? It's a couple slides back. Chitin. That's just a reminder, that was a couple cells back. But fungal cells come in 2 basic forms. We have yeast fungus and we have mold fungus. And the difference between those is the number of cells. So unicellular funguses are called yeasts. Okay, they are single-celled, unicellular. That means that their whole body, the whole organism is made up of one single cell. And the molds, and sometimes they're called hyphae. I will never call it this, but I think I saw it somewhere in your textbook, them referring to a fungus as a hyphae, which I thought was weird. I will always refer to it as a mold, but a mold is a multicellular fungus, meaning it has multiple cells as part of one singular organism. And the thing to remember here is that yeasts reproduce by budding. So they reproduce similar to bacteria. They basically just have one cell, and that buds into 2 cells. And then that buds into 4 cells. And then those will further bud off. So you have one cell that just produces a duplicate of itself, and it buds off. Okay. And then molds reproduce by spore production. So molds basically produce, it's kind of like seed dispersal, like plants produce seeds. Molds produce lots and lots of individual spores, which are reproductive structures, and those spores can then distribute into the air. And then wherever one of those spores lands, a new mold will grow. Okay, kind of like a seed or something like that. Something called a dimorphic fungi. So again, look at the word. Di means two. Morphic means like forms or morphologies. So dimorphic fungi are actually capable of doing both. And they can be a yeast or a mold depending on the temperature. So they are a yeast or a mold depending on the temperature. Okay. And we'll talk more about dimorphic fungi when we get to the infections later, but dimorphic fungi are some of the most critically important infections. They cause some of the most serious infections, and some of the most deadly fungal infections are caused by dimorphic fungi. And these are just some examples here, just to, I think, this is a yeast from somebody's urine, right? Vaginal yeast infections, oral yeast infections, dogs get ear yeast infections, right? Those are very, very common infections, yeast infections. And then this is an example of a mold. You can see it's got multiple cells. It's a much bigger structure. Molds can cause things like penicillium. A lot of people are allergic to molds because they breathe in the spores. They're very, very allergic to them. But they can also cause all kinds of infections, which we'll talk about later. Now, something that is kind of important about funguses is that funguses, and what kind of sets them apart from some of the other eukaryotes, is that they are all heterotrophs. Now, what heterotroph means is the heterotrophs of the world are what we call the consumers. And these are organisms that cannot and do not produce their own food. Okay? So they get all of their food from another organism. They have to consume something else in order to produce their own food. Now, we as humans, we're heterotrophic too, right? I can't produce my own food by sitting in the sunlight. I wish I could, but I can't. I have to eat actual food. I have to eat plants or animals or funguses in order to survive. That is how my body produces energy. And the same thing is true for funguses. All funguses are heterotrophic. But where or the type of organisms that they consume is going to depend on the organism. So there are a lot of funguses, and this is kind of what we know funguses to be. It are saprobes. And what saprobes mean is that they feed on dead or decaying organisms. That is a saprobe. It's a heterotroph, so an organism that has to eat something else to produce energy, but the organism that it's eating is dead or decaying. Okay, and this is kind of the classic fungus, right? Mushrooms grow on the forest floor from all the dead leaves and matter and animals that have died in the forest. They get their energy from the soil and the decaying leaves and the decaying rot. Or they get, this is a picture of a fungus growing on a fallen tree. Okay, or you get mold growing on the loaf of bread on your counter, or an orange that has gone mushy. Right? That is dead or decaying organism. Okay. An orange that has been picked from a tree is no longer living. It is something that is dead and it is rotting. Okay. A parasite is just the opposite. It's a heterotroph that feeds on living tissue. And these are the organisms, the parasitic fungi are the ones that are going to cause human disease. And that's why they cause disease, because they are feeding on our human live tissue. And that's why they harm us. That's why they cause disease. But the truth is that most fungi are actually going to consume and live on whatever they are able to. There are some funguses that only feed on human tissue. They only survive by living in humans. But most funguses are going to be capable of growing most places. But if they come in contact with a human, sure, they'll grow there. It's a great place. It's warm in there. But they don't need humans to survive. So that's kind of a distinct difference, I guess, that we will explore further. But for now, that's the difference between a saprobe and a parasite. A saprobe feeds on dead or decaying tissue. A parasite feeds on living tissue. All right, we're going to skip this for now. Okay, next we have the protozoa. Now, the protozoans are kind of an informal group of organisms that don't really fit in anywhere else. So they are eukaryotic organisms that are quite literally not plants, animals, or funguses. And so they just get thrown in this other category called protozoa. If it's a eukaryotic organism and it's not a plant, an animal, or a fungus, it's a protozoa. It's just kind of the extra group. And so because of that, there's not a lot of characteristics that they all share. A lot of protozoa have different characteristics. They look very different from each other. Some of the things that we can say is that most protozoa, not all, but most are heterotrophic and unicellular. Most. So most of our protozoa are going to consume other organisms for food. They are heterotrophic, and most of them are unicellular, meaning that the whole organism is one cell big. Now again, that's not 100% true. That's why I wrote most. A lot of them can either be saprobes or parasites, just like the funguses, and the same rules apply there. Some common protozoan infections that you may recognize, I don't know if I have it written down here. This one right here, this guy causes giardia, which is also known as traveler's diarrhea. If you've ever been to a foreign country and drank the water and ended up horrifically ill for like 2 weeks, you probably had giardia. Or if you even just drank water in the forest that a deer pooped in, you probably had giardia. Another one is this guy right here. You don't have to know these names, but you have to know these two diseases that I'm saying. Okay, so giardia and malaria. Okay, malaria is an incredibly significant disease. We'll talk way more about it later. But for now, you have to know that it's caused by a protozoa. Malaria is not caused by a bacteria. It is not caused by a virus. It is caused by a protozoa. And then our old friend, I think we've talked about him a few times, the Trichomonas vaginalis causes that trichomoniasis, the vaginal infection. Okay. Now, I put this up here. You do not have to memorize this picture. That's not the point of this. The whole point of this slide is just to show you that most protozoa have very complex life cycles with many, many stages. Even things like malaria takes place in at least 2 hosts, right? It has to be in a mosquito and it has to be in a human. Within a human, it visits at least 2 organ systems. It has to be in your liver and then in your red blood cells at some point. It requires a lot of moving parts, and different things happen at different times inside different hosts, and all of those things have to come together for this to be able to cause infection. So protozoan infections are very complex and very difficult to treat because they are so complex. And protozoans in general, they do reproduce both sexually and asexually. For malaria, there are parts of within its individual cycle where it reproduces sexually, and then other parts of its life cycle where it reproduces asexually, which means without sex. Right? You put that A in front of it, means without sex. Okay? And if you don't know what sexual reproduction is, I hope all of you know what it is, but if you don't know what sexual reproduction is, it is just the process of it requiring 2 organisms to make a new one. Okay? It requires a parent with X chromosomes usually and a parent with Y chromosomes that combine to form a new organism, rather than being able to create an organism by yourself, which is what asexual reproduction is. Like budding is asexual reproduction. Okay. And then our last group are the helminths, and the helminths are a group of worms. that can infect humans and other animals. And it's important to keep in mind that helminths are worms, which means they are animals. And they have a lot of the same characteristics that other animals have. And we'll talk about more of that in a second. They are multicellular, right? They're animals. They're multicellular. They have reproductive tracts. And they have digestive tracts. Okay, all things that are common in all animals, okay? What an animal's reproductive and digestive tract looks like is going to depend on the animal, but all animals have them, okay? And worms are no exception. Similar to protozoa, many helminths have very complex life cycles that include the spreading of eggs and the growing of larvae that then get ingested by something else. What was I trying to talk about? Sorry. So something to keep in mind about helminths that some people get confused about, and I think we touched on this a little bit, is these are worms, right? Some of these worms, if anybody's ever seen a video of a tapeworms can be like 50 feet long. That's most certainly not microscopic, right? I can see a 50-foot long worm. It's not microscopic. But the thing about helminths is that most of the time, helminthic infections happen because somebody consumed eggs, and the eggs are microscopic, right? Nobody gets a tapeworm because they swallowed a 35-foot worm, right? That's not how that happens. You get a tapeworm because you swallowed a microscopic egg, which then grew into a 35-foot worm. Okay, so this is some pictures of the helminth eggs. We don't have to know the differences between them. They're just some pictures so you can see. And this is just a picture of some of the different types. You don't have to know those right now. It's just an example. Okay. But that is why helminths are kind of included in microbiology, is because their infective stages, how most people get these worms inside their body, is microscopic, even if the worms themselves are not. So again, helminths are animals. They reproduce sexually. Okay, it takes a boy worm and a girl worm to combine and create a new organism. Now, the thing that's actually kind of fun is that a lot of these worms are what we call true hermaphrodites. Now, I know hermaphrodite is a really non-PC term when you apply it to humans, but in science, it has a very specific definition. And these worms are true hermaphrodites, some of them, which means that they produce, within a single organism, they produce both eggs and sperm. So they have structures where they basically sexually reproduce with themselves. Okay, they produce eggs in one part of their body, they produce sperm in another, and that eggs and sperm meet inside of the body and produce new organisms. So it's a subtle difference between asexual reproduction and hermaphroditic reproduction. Because in asexual reproduction, there's no combining of eggs and sperm. Okay? This is sexual reproduction, it's just happening within a single organism, which is very bizarre and very cool. But yeah, you can see this is the inside of a certain type of worm, and they're producing eggs, and they have a vulva and a uterus, but they also produce sperm. Okay, so something similar to like a vas deferens, where they produce sperm. So they fertilize themselves. Okay, and the most common, this is the only example we're going to talk about right now, is Enterobius vermicularis. Okay, and you have to know its name. You don't have to know how to spell it, but you have to recognize it. Enterobius vermicularis is the most common helminth infection. I can't spell today. In the U.S. Okay. Enterobius vermicularis causes the most common helminthic infection in the United States. And the infection, does anybody know what this infection is called? Has anybody had it? Nobody's going to want to tell me if they had it. It's okay. It's called pinworms. Pinworms is the infection. Enterobius vermicularis is the organism that causes it. And it's very common. It is most common in children. And the way that it is spread, it's a kind of crazy infection. That's why I always like to talk about it, because it's kind of fun. Is that pinworms spread through the digestive tract, just like a lot of other worms. But in particular, the adult female worms specifically lay their eggs in one place on the human body. And that's the only place that they will lay eggs. And they specifically lay eggs in the anal folds, which is just a fancy way of saying on the outside of the butthole, okay? It's on the outside of the anal opening. And so what happens, and why this spreads so quickly through children, is because at night, when the host is nice and relaxed and not moving and not disturbing them in any way, the female worms will crawl out of the anal opening. They live in the digestive tract. They crawl out of the anal opening, and they're very small little worms. They're a couple millimeters big, and they lay eggs in the anal opening. And the process of the worm crawling out of the anal opening and laying eggs is very, very itchy. And kids will scratch their butt, and kids have no problem scratching their butts and then sticking their fingers in their friends' mouths. Right? Kids are disgusting. It's one of my favorite things to say. Children are gross. My son, his favorite activity used to be licking the floor. I had to literally wrestle him through an airport one time because all he wanted to do was lick the floor. Buddy, I don't know what's up, but he just wanted to lick the floor all the time. And so kids, what will happen is they scratch the anal opening because it itches, specifically at night, and then they will pass it on to they'll touch something else, and then the other kid is going to put the toy in their hands, or they've just shoved their hands into an adult's mouth, or whatever else. Anybody who's ever had a newborn or been around one, they will shove their hands anywhere. Okay, and that is why it's so common. I actually when my son was little, I had a little girl that I drove to work. Her parents worked at Tech 2, and I drove both of our kids to work. And Ella was the girl's name, and she was sick for a week one time, and so I didn't drive her to school. And her mom was kind of like being kind of, not shady, but she was like, oh yeah, Ella's sick, and she sent her back to school, and her mom was trying not to tell me what had happened, and then the first day back in the car, Ella was like, I had worms in my butt. So, I think your mom didn't want me to know that, but thank you, Ella, for the information. But pinworms are very common. They spread through daycares and kindergartens and things like that very, very quickly. So if you ever have a kid one day and they start scratching their butt, specifically at night, it's very possible that they have pinworms. Most common infection in the US. Okay? All right, now we're going to move on to chapter 5, and we're just going to get through as much of this as possible. Again, I am going to be skipping some slides. Just keep up with me and mark out whatever I mark out. All right? Now we're talking about, chapter 5 talks about viruses and prions. Now, viruses and prions collectively are what we call acellular. That's not how you spell cellular. Acellular organisms. Okay, and we call them acellular. Some people don't even call them organisms. They're acellular particles to some people. But what that means is you put the A in front of it, and that means without. So without cells, acellular. Right, so viruses and prions do not have cells. They're not made up of cells. We're going to talk about what they are made out of. They don't have cells. The birth of virology, so how viruses were first discovered. Viruses, in general, is a unique group of what we will call, from now on, biological entities. Okay, we don't necessarily call them organisms because they don't have cells and they're not alive. So they're biological entities known to infect every type of cellular organism on the planet. Now, to be specific here, I don't mean that a single virus can infect all other cells. What I do mean is that for every cellular organism that exists, there is a virus that infects it, at least one. All plants, bacteria, funguses, humans, animals, everything on this planet that is composed of cells can be infected by a certain number of organisms. Okay, there's not a single one that is left out. Okay, and they are extremely abundant in nearly every environment on Earth. It's hard to conceptualize just how many viral particles exist in the world. But again, I'm going to give you some numbers. You don't have to memorize these, but it's estimated that every milliliter or a quarter teaspoon of water, a quarter teaspoon or 1 ml of water, contains about 10 million viral particles. 10 million in a quarter teaspoon of seawater. So yeah, it's really hard for our brains to really conceptualize how many that is and how they even physically fit, because we're still picturing things that are the size that we can imagine. It's really hard for our brains to even think about something that's small enough to fit in that amount of water, but it's true. And a study done in Spain collected viruses like you would rainwater. So they put out little collection things. They just put them out in the open and collected viruses like you would rainwater. And they found that 800 million viral particles fell out of the sky onto every square meter of Earth every single day. Just fell out of the sky. Okay, viruses can survive in the air. They can float in the air for days at a time. They have so little mass that they just float in the air. Or they can be transmitted through touch or anything else. They're everywhere. The first virus ever discovered was the tobacco mosaic virus, and that was discovered in 1892. Okay, so just to give you kind of an idea, remember that the beginning of bacteriology was around the early 1800s. So it was about 100 years after bacteria were discovered before somebody figured out that there was something even smaller. However, it does require a scanning electron microscope to view a virus. Okay, remember we talked about that in chapter 2, that to view a virus, you have to have a scanning electron microscope. And those weren't invented until the 1930s. So the first virus was not actually seen until the 1930s, even though they were named and discovered before that. And we'll talk more about this later, but some of the most important viral vaccines, and we're going to talk about vaccines a lot in this class. I know they're a hot topic right now, but we're going to talk about them a lot. Vaccines were not developed really until after the scanning electron microscope became popular. There were a few vaccines that were developed before that, but until viruses could be seen and isolated and grown, a lot of these vaccines were not possible. Okay. So viruses as pathogens. Viruses are what we call obligate intracellular pathogens. And all that breaks down to, again, look at the word. Pathogens, meaning they can cause disease. Intra means inside. So intracellular means in a cell. And obligate means it has to. So an obligate intracellular pathogen is a pathogen that has to be inside of a cell to be a pathogen. Okay? Viruses require a host to reproduce. Viruses are only spread and able to make more copies of themselves if they have a host. They cannot do it outside of a host. And viruses have caused some of the deadliest epidemics and pandemics throughout human history. Things like influenza, the 1918 flu pandemic was immeasurably awful to human society. Ebola, COVID-19, right? You all lived through that. We all know how awful that was. Those are all caused by viruses. And we're going to talk about that more as we go through, but we're going to touch on it now. But treatments and cures are very, very difficult for viruses, right? And most of us have been there, right? You go to the doctor, they tell you that you have a cold, even something mild, like you have a cold, or you have a flu, or whatever. What do they tell you to do? They tell you to go home, take some Sudafed, and go to sleep. That's it. That's the cure, right? There's no cure for it. There's no antibiotics. There's not really very many medicines that can be given to help you feel better. Viruses are very, very difficult to treat. And the reason for that is because of the simple fact that when a virus is growing inside of our body, it is inside of our cells. So how are you supposed to treat something that's growing inside of your own cells without killing off your own body? And so there are some viral infections, things like hepatitis C and HIV, that do have treatments. Those treatments are very, very upsetting. I'm trying to think of the word, but they have a lot of side effects. Hepatitis C treatment is like being treated for cancer. Okay? It destroys a lot of your own cells. It makes the host very, very sick. Treatments and cures for viruses are very, very difficult. Even at the height of the COVID-19 pandemic, right? There were all these like treatments that were put out, and you can get like Tamiflu. I don't know if anybody's ever taken Tamiflu, but Tamiflu, even as a relatively easy thing to get, if you have the flu, you can take Tamiflu. I've never taken it, but I've heard that people who do take it, sometimes they're like, the Tamiflu was worse than the flu. It made me so sick. It made me have diarrhea and have vomiting and things like that. Like, this is worse than just fighting the fever for 2 more days. So treatments are hard, and a lot of times they're not effective. Tamiflu has to be taken within like the first 24 hours of infection, and even then it's only like half effective. Okay? And it's all because of the way that viruses grow, that they only grow inside of a host cell. And it's true that for most viral infections, vaccines remain the best course of action. The best course of action and the best type of, not treatment, but cure for a viral infection is to not get it in the first place. Okay? To stop your body from getting it in the first place. All right, so viruses. We are going to talk about viral cells or viral particles. I slip up sometimes too. We don't call them cells because they're not made of cells. They are particles. Okay, they're not made of cells. So all those rules about what we talked about when we talked about prokaryotic and eukaryotic cells, none of those rules apply because we don't have cells. At their core, I'm going to show you exactly what's inside of a virus in just a second, but at the base level of what they are, they're a small bit of genetic material wrapped in protein, and that's it. They don't have any of the other things that makes up a cell, but they have very, very simple structures. Now, just to show you some of the size comparisons, so this is obviously blown up. This is not a true to size diagram, but this big blue thing here, this is a yeast cell. Okay, so that's a eukaryotic cell. And this is an E. coli, which is a bacteria. Okay, viruses, even the largest virus is a little bit smaller than the average bacteria, but the average virus is going to fall around here somewhere. Okay, so this is an average virus. They are incredibly small. They're usually between 10 and 100 times smaller than even a bacterial cell. Okay, so on the outside of a virus, the viral covering, all viruses have what's called a protein coat, sometimes called a capsid, but it's just a layer of protein that circles around the outside of this particle. So all viruses have some kind of protein coat. Some viruses have what's called an envelope, which is an extra layer of protein that goes around the outside of the capsid, okay, like you can see here. This is a naked virus, so one that does not have an envelope. And yes, they really call them naked viruses. It doesn't have an envelope, and this one does have an envelope. Okay, it's just an extra layer of protein on the outside of the capsid. Some of them have it, some of them don't. And some of them have attachment structures, some of them don't. Attachment structures are things that help them attach to their host. It's exactly what it sounds like. So if you've ever heard of something like COVID-19 has spike proteins. That's how it attaches to the human host. That's what a lot of the vaccines are treated against. So that word, the COVID-19 spike protein, is kind of a buzzword that you hear about the vaccines. Okay. And this is just some pictures. Again, you don't have to know the differences of this, but it's just to show you that a lot of these viruses look very different. Okay, some of them have weird little spikes sticking out. Some of them are in like coils and things like that. Some of them have these funny looking leg things. Some of them are perfectly round. Some of them look like hexagons, right? They have lots and lots of different structures. It's just important to remember that all of them have a protein coat. Some of them have an envelope, and some of them have attachment structures. Okay, so that's the outside of the virus. The inside, or the viral core, all viruses have DNA or RNA. They never have both, right? The vast majority of cells, I can't think of a single example of one that only has one or the other, but I'm gonna say the vast majority of prokaryotic, eukaryotic cells have both DNA and RNA. Sometimes lots of different kinds of DNA and RNA. Viruses have one or the other, never both. Now that DNA or RNA can be double or single-stranded, which is unlike typical cells, right? In cells, DNA is double-stranded, RNA is single-stranded, but in viruses, it can go either way. So you can have double-stranded DNA, you can have single-stranded DNA, double-stranded RNA, or single-stranded RNA. Okay. Some viruses have extra enzymes or protein for aid in infection, but not all of them. We'll talk more about that later. All right, but you can see, again, just some examples here. This is a helical vaccine, a helical virus that only has RNA. And so you can see the different shapes and the different types of genetic material that they have. Again, they have DNA or RNA. They never have both. Because viruses are not considered alive, right? All of those things that are required to make up a cell, viruses have almost none of those things. They have a little bit of genetic material, but not enough. They don't have ribosomes, they don't have a cell membrane, and they don't have cytoplasm. They don't have any of those things. So because they're not made out of cells and they're not considered alive, you are not ever going to find them on a phylogenetic tree. So all those trees that I showed you that had bacteria, archaea, eukarya, viruses are not anywhere on there because they're not alive. That is a tree of life. Viruses don't exist on that tree of life. But a lot of viruses are classified based on three things: whether their genome is DNA or RNA, whether their genome is single-stranded or double-stranded, and whether or not the virion is enveloped or not. Okay, so we tend to group these viruses based on those three categories. So for example, these in this box, and you don't have to memorize these names, but these in this box are enveloped and have double-stranded DNA. These over here are non-enveloped and have double-stranded DNA, right? We kind of clump them together with viruses that have similar structures based on what type of genetic material they have, DNA or RNA, whether it is single-stranded or double-stranded, and whether or not they have an envelope. All right, now we're going to talk about how viruses replicate. So if viruses don't have any ribosomes, right, we didn't talk about ribosomes at all because viruses don't have any. How do they replicate? Okay. And the general gist of it is that viruses infiltrate their host cell and use the host cell to reproduce. And this is a picture of this kind of outline right here that I'm doing in orange. You can't tell, used to be a bacterial cell. And all of these other things, these funny looking guys right here, these are all viruses. And this virus has infected this cell and it's using it to reproduce itself, and all of the new viruses are busting out of the bacterial cell. So this virus infiltrated this bacterial cell, it used all of its machinery, it stole its ribosomes, it stole all of its machinery to synthesize new copies of itself. Okay? In general, viral replication has 5 steps. And the 5 steps of viral replication are relatively intuitive. Hopefully this all makes sense. It shouldn't be anything kind of crazy. The first step to replication is going to be attachment. So step 1, attachment. What happens during attachment? The virus somehow comes in contact and attaches to its host cell. Okay? How that happens is going to depend on the type of virus and the host cell. There are lots of different ways this happens, but attachment is just the process of attaching. Imagine that. Right? Pretty easy. But it comes in contact with its host cell and it attaches to the outside. Next, we have entry. So after coming in contact with its host cell, the virus somehow has to get inside. And again, how this happens is going to depend on the virus and the host. There are some hosts some viruses that trick the host into bringing it in, like they make the cell think that it's some kind of food source, and they bring it in like a Trojan horse kind of thing. Some viruses poke holes in the cell and force their way in. It just depends on the virus, but some kind of entry. The next step is going to be the virus taking over the host cell machinery and replicating itself. Okay, so it is going to remember that a virus is made up of a little bit of protein and a little bit of genetic material, and that's it. So it has to make some copies of those 2 things. It's going to make copies of its protein, copies of its genetic material, and make more copies of that. It's going to steal its host's ribosomes and its host machinery and enzymes, which we'll talk more about later, to make more copies of itself. Okay. Step 4 is assembly. So those little pieces of protein and those little pieces of DNA are going to get assembled into a viral particle. Right? The protein is going to wrap around the genetic material. It's going to assemble the way it needs to be and create new viral particles. And then the last step is release. And it's just what it sounds like. The newly made viruses, are then going to escape their host cell. Now, the release may or may not cause what's known as lysis. And lysis is a cell bursting open. Okay, so some viruses, when they are released from their hosts, they burst open their host cell, killing it. Right? They burst it open, they kill the host cell. I gotta say, it's really funny to watch all of you tilting your head. Sorry I wrote it sideways, but you all are tilting your head in the same way. It's really funny. But they basically burst the cell open, killing the cell. There are some viruses that are extra sneaky, and they actually don't kill their hosts. The viruses can hang out inside the host for long periods of time without actually killing it, which is arguably more dangerous because they can stay in there for months or years, causing symptoms over long periods of time. Again, it depends on the virus and the host. Okay. Now, this is the basis of viral replication. This is some version of this happens with basically every virus that exists. Now, something that can happen, but doesn't always happen, okay? So this basically always happens. Replication, this is a blueprint for basically what happens with every virus. Something that can happen, but doesn't always, so it can happen, but doesn't always, and it happens with specific viral types, is something called integration. And what integration is it's the integration of the viral DNA into the host DNA. Okay, and so all you really need to focus on here is just this viral DNA inserting itself and integrating into the host DNA. And what this will cause is a couple of different things that we're going to talk about in a second. This DNA becomes a part of the host now. So this host cell has been changed forever. Okay, that viral DNA is now integrated into the host genome. And a lot of the most destructive viral infections are capable of doing this. Things like HIV, HPV, herpes, right? Those are the things that stay around for a long period of time, right? You guys know that if you get herpes, you have herpes forever. If you get cold sores once, you will have them for the rest of your life. And that is because of integration. Okay, this virus has now made itself permanently a part of your human host body. And it's very difficult, if not impossible, to get rid of. Okay, now because of that, those two different things, there are a couple of different types of viral infections, kind of different categories of viral infections. 3, in fact. Okay, so the first type of viral infection is called an acute viral infection. And acute usually means quick, it's, fast and furious, quick and severe usually. So these are infections that happen quickly and then they're over quickly. You get symptoms, they suck, you feel awful, and then they go away. Okay? They are short-lived, very quick, short-lived viral infections. And in the case of acute viral infections, the viral genome is not integrated. There is no integration in acute viral infections. In acute viral infections, your human body gets sick, gets infected with a virus, and then your immune system kicks it out in a couple of days. Okay? So these are things like the flu, COVID-19, norovirus. Anybody's had the stomach bug in the last couple of weeks? I feel like it's going around. Things like the measles is usually hand, foot, and mouth disease, right? There's all kinds of different viruses that are acute. You have symptoms that go away. Okay? Simple enough. The next type are called latent viral infections. And in latent viral infections, what happens is that viral DNA is integrated into the host genome and then basically goes to sleep. It remains dormant. So it integrates itself into the host and then goes to sleep. But because it's asleep, it's still there. And at any time, it's basically asleep. At any time, the virus can wake up and cause symptoms. Now, the big key here is that while the virus is asleep, you don't have any symptoms. It's asleep. It's gone to bed in the cell. And it's not causing any problems. But at any time, the virus can wake up and cause symptoms. The most common example of this is the herpes simplex virus, also known as HSV, that causes genital and oral herpes. Anybody who has cold sores know that you get them at random times. They're called cold sores because a lot of times you get them when you're sick. Okay? So you get infected once, it causes symptoms. Like I got cold sores, which is a fancy, we call them cold sores to make it sound less bad, but it's the same thing, right? Okay, it's oral herpes. I got cold sores because I stole my brother's soda when he was like 15, and I would have been like 7. I stole his soda, he had a cold sore, so I got a cold sore. And now I get them my whole life, right? And I usually go, as I've gotten older, more time will go between getting cold sores. I haven't had one in like 2 or 3 years now, I guess. But I used to get them every couple of months. But when I don't have a cold sore, I don't have any symptoms, I can't spread it to other people, I'm not contagious, that virus is just asleep. It's not doing anything. No symptoms. Another example is the varicella zoster virus, which causes chickenpox and shingles. So when you get, and I know most of you did not have chickenpox, you guys are kind of the first age group, ish. I was like the last people who got the chickenpox because the vaccine for chickenpox didn't become available until like 5 years after I had it. I was like in middle school when the vaccine became available. But if you did get chickenpox sometimes you get chickenpox, you get these itchy spots, you're itchy, you have a fever for a couple of days, and then it goes away. Sometimes the chickenpox virus will go to sleep inside of that person. And then 40 years later, it reemerges as shingles, which is a virus rash that usually happens on the trunk. It's supposed to be exceedingly painful, so not looking forward to that if that ever happens. But that is an example that chickenpox virus goes to sleep for literal decades for most people. And then anytime, it's usually when a person reaches kind of an advanced age and their immune system starts to go down, that it will reemerge as shingles. Now, the third type are chronic viral infections. Now, the difference between latent and chronic is in chronic, we have integration of the host genome and then continuous replication for long periods of time. Okay. And this usually results in continuous and worsening symptoms. So instead of the symptoms going to sleep and having no symptoms and being not contagious until you have a reemergence, chronic infections will get worse over time, and you consistently have symptoms over many, many years. A really, really common example of this is the human immunodeficiency virus, also known as HIV. The way HIV works is when you're initially infected with HIV, you usually have very mild flu-like symptoms. You probably wouldn't even recognize that it's anything serious. And then it will stay replicating in your body for decades. And HIV is really sneaky. It stays around for a long period of time, and it causes minimal symptoms, but you will start to have things like you'll get fevers every once in a while. You'll get like weird rashes. There's a lot of symptoms that go along with HIV. But those symptoms will be really, really subtle for a long time. But what happens after decades and decades and decades and decades is eventually this virus completely shuts down your entire immune system. So what it's been doing for decades is it has been continuously replicating and systematically invading all parts of the immune system and shutting them down one at a time for years and years and years and years. And eventually it causes a disease called acquired immunodeficiency syndrome, also known as AIDS, which is basically where the person no longer has an immune system. Another one is HPV. We'll talk about HPV in much more greater detail later, but HPV is a virus that's known to cause cervical cancer. And it's a sexually transmitted infection. It infects the cervical cells, but it also infects the urethral cells, the vas deferens, the testes, the ovaries, the uterus, everything involved with genital tracts. And it will stay there for decades. And in a significant portion of people, the cells over time will cause cancer to happen. We'll talk more about that later. All right, so we're going to talk quickly about viral cultivation. Viruses require special cultivation techniques because they won't be active without a host. Remember that viruses require a host to grow. So bacteria, you can slap them on a plate and put them in an incubator and have tons of bacteria overnight, right? You could go home and grow bacteria if you wanted to, right? Viruses, not so much. Viruses require a live host cell to grow. They cannot be cultured on typical media. They require a host to grow. The most common techniques for viral cultivation are things where you grow some type of animal or organism cell. Live animal inoculation, there are places that for certain viruses, the best way to get more virus is to infect a live animal. Mice and rats are obviously the most common choice, but sometimes monkeys and things like that will be injected with viruses so that it grows inside their bodies, and then we can harvest the viruses from their bodies. You can also do bird embryo inoculation. This is actually how flu vaccines are made. Flu vaccines in the United States, and mostly across the country, across the world, are made inside embryonic eggs. Bird eggs, chicken eggs. So they take chicken eggs that have been fertilized and have an embryonic chicken in them, and they inject that embryo with flu virus. and that flu virus will grow over a couple of weeks, and then the flu virus is harvested from the eggs, and then the vaccine is made from that virus that is harvested. It's actually a pretty common thing that if you have a severe egg allergy, you can't get the flu vaccine or a traditional flu vaccine. There are other types that are available. But this is why it takes flu vaccines. So I don't know if you guys know this, but they start making the flu vaccines for next year almost a year ahead of time. So we are smack dab in the middle of flu season right now. Probably in January, they'll start making and producing the flu vaccines for next October. Okay, they have to be made many months in advance because this process takes a very long time. Another way that this can be done is through something called cell culture. And what cell culture is keeping human cells basically in liquid media. And then those human cells can then be inoculated with virus, and the virus will grow inside of the human cells. Now, the tricky part about cell culture, and we're going to talk about a specific case in just a second. We're going to finish, I know it. Okay, so the tricky part about cell culture is that if I took some of my cells right now, my normal cells, I swabbed them out of my mouth, and put them in all the right media, in all the right incubators, and did all of the processes that were required to grow cells, they would stay alive for a couple of weeks tops, and then they would die. Because your human cells are not meant to stay alive forever. Your human cells are not immortal. What's the saying that like you are a new person every 7 years or something? Because all your skin cells get replaced. All your heart cells get replaced. Everything in your body gets replaced on a time schedule. Your cells do not live forever, even if they are in the perfect media. Now, there is an exception to that, or several exceptions. But the first exception to this, the reason that cell culture was developed, how it was developed, is through something called HeLa cells. Now, HeLa cells is an immortal cell line. This was a cell line that was discovered inside of a woman named Henrietta Lacks. Okay? That's why they named them HeLa cells. They named them after her first and last name. They are a cell line that stay alive forever. But the reason that they found these is because Henrietta Lacks had a very, very aggressive form of cancer. And her cells were first discovered in 1951, and her cells are still alive. Okay? That's why she had cancer. Your cells are not supposed to survive forever. If they survived forever, it's abnormal, and it's going to make you sick. That's called a tumor. That's what a tumor is. So HeLa cells, we're going to talk about how they were discovered more in just a second, but HeLa cells are used to grow viruses for research and vaccine production and countless other ways. Millions of researchers and companies use HeLa cells every single day. It is incredibly I can't even explain to you the breadth of HeLa cells. The things that have been discovered because of these cell lines, it's impossible to put on a list. The world that we know as it today would not exist without this cell line. Now this woman, I think it's important to talk about this kind of stuff. It's really uncomfortable. It's hard to hear about sometimes. But this woman was a poor black woman in 1951, and her doctor stole something from her. He didn't tell her that he was taking her cells. He collected her cells from her cervix. She had cervical cancer. He collected her cells and then used them to further his own research. And he made a lot of money off of her. Never told her, didn't tell her family. She died about 3 months after her cells were taken. And they're still used today to make billions of dollars. And to this day, in 2025, her estate has never been compensated. Now, this is extremely illegal, right? Nobody can take anything from your body without your express permission. If you got caught doing this, it would be extremely illegal, and you would be immediately sued. However, at the time, it wasn't illegal because nobody knew that it was possible. So there wasn't a law against it. So was the person who did it breaking a law? Was he breaking a moral obligation? And then looking at it now, if, I think that a lot of us could be like, well, yeah, obviously her family should be compensated, right? But who's supposed to compensate her? It's not like Gila cells are owned by a single company. Gila cells are basically public. You could go online and buy them right now. There's not a single entity that owns them. So who should have to pay her estate? The entire United States? Everybody who's ever used them? The ethics of the situation are very complicated. But it's talked about a lot in this book. It's The Immortal Life of Henrietta Lacks. It's a really interesting read. And it really gets into medical ethics and personal ethics, and scientific ethics. And I think it's just important to think about things like this, because this was not that long ago, right? My dad was born in 1955. So my dad's still alive. This was not that long ago. And this was very normal. So what things are we going to look at in 50 years and be like, oh my God, I cannot believe that somebody did that? And it's important to think about those things and how poor black women can be taken advantage of by rich white doctors and have money made off of them. It's just important to think about, even if it's a little uncomfortable. All right, so that's all the material. We're just going to skip the next 2 slides. I think it was prions. Just skip them. We won't talk about it.